All right, hey, thanks for coming to today's version of Micro Seminar. It's the first one in a while, and I just want to give a plug. If anybody's out there is interested in giving a Micro Seminar, just please contact either me, Dave Baltris, Lizzie Wilbanks, or Cameron Thrash. We're trying to schedule things for kind of April on, and so ton of stuff open. Just please contact us. We'd love to host you and love to have you share your science with us. So it's my absolute pleasure today to actually introduce Dr. Catherine Milligan-Myhre. Myhre, is that it's
yeah,
Myhre? All right. So first, I want to just give you a huge thank you from all of us, because you've been doing some amazing work the last couple of days, just kind of internalizing and giving out a lot of support to a lot of grad students out there. So just for me, and I think from a lot of people, just thank you. I'll link to the thread, and I'll link to the all of the advice and all of the situations that you've shown. I'll link that in the YouTube comments. And so if anybody hasn't taken a look at that, just please take a look in terms of grad school awareness and grad student awareness. It's really important to kind of support people, and Kat's done a really awesome job relaying people's stories over the last few days. All right, so now that I've said that, it is my absolute pleasure to introduce Dr. Catherine Milligan-Myhre. Kat was an undergrad and a grad student at the University of Wisconsin-Madison in Laura Knowles' lab for graduate school. And then we actually share a background. So Kat was then a postdoc in Karen Gilliman's lab. Karen Gilliman was one of my PhD advisors. And Kat was also co-advised by Bill Kresko at Oregon. So Bill was actually my first rotation in grad school was under Bill. So I was working with Sticklebacks way back when. And so it's awesome to see kind of how the system has blossomed and thrived over the last few years, since 20 years or so since I was actually working in it. Today, I think Kat's going to talk about sticklebacks and microbes. There's a lot going on here. I got bee guys outside and stuff. There's a lot of activity. So, but aside from that, please take it away, Kat, and we're looking forward to hear what you have to say.
Thank you, Kuyana. So, ubla lotak, ubana ataga napaktak mi runa, kikitagak mi runa, nipak mi runa. Apaga James McClellan, Anaga Sally McClellan. So if you see them, tell my parents, Sally and James McClellan, you heard me on the web. They'd be super psyched. So what I said is, ubla lotak, good morning, good day. Inipak mi aruna, I'm Inipak, Kitiktagruk mi aruna, I'm from Kotzebue, Napaktuk mi aruna. My people are from the Napaktuk Mi'kmaq tribe in Northwest Alaska. And I'm currently, as Dave said, an assistant professor at the University of Alaska Anchorage, and I've been here for 3.5 years. So I'm going to do a quick share, which of course I knew how to do a minute ago. Here we go. Entire screen. You're going to see yourselves for a second. Let's see, are we sharing?
Not sharing yet.
Okay, there we go. Okay, so now you should see stuff. So what I'm going to, I want to start with saying land acknowledgement. So thank you to the Denai people whose land the University of Alaska is on and who are still here. So just want to acknowledge those people. So as Dave said, I'm going to be telling you about the work that I do adapting an evolutionary model for host microbe studies. And this is work that I got into as a postdoctoral fellow at the University of Alaska Anchorage. And I was really lucky. So when I was a grad student, I studied pathogenic microbes, and I did that as an undergrad as well. And so I had all this experience in microbes that can do harm. And that's what a lot of people were studying for a really long time. We've been studying microbiology for hundreds of years, but it's always been in terms, for a long time, it's historically been in terms of how are microbes damaging us. And that was really important when I was an undergrad and when I was a grad student. But towards the end of my grad program, we started thinking about not just how we can conquer these microbes, how we can kill these microbes, but also how we benefit from microbes. And it turns out that these microbes are not just trying to kill us. It's not all microbes are trying to kill you. And in fact, a lot of microbes live symbiotically with us. They don't really know what we are, right? But they live with us, on us, all over our bodies. They're on our skin, they're in our eyes, they're underneath our armpits. They're part of the reason our armpits smell. You know, they're just a major part of our body. And these microbes are not actually trying to harm us. They're not trying to force our cells to break open so they can extract nutrients. They're not producing toxins to kill us. But instead, what they're doing is really great things for our body. They're stimulating our immune system. They're stimulating our growth. They're stimulating brain development. You know, these little tiny microbes that are associated with your body at some point in your lifetime stimulated a cell which sent a signal all the way up to your brain to say, hey, you need to kick in and start developing because we need this host to survive. So not saying that the microbes thought that, but the result of all these interactions help stimulate our development and our growth. And they also do things like make things that we can't make, like vitamin K. Now in our lab, we're specifically interested in the microbes in the gut. I really like the microbes in the gut. All the things that I just told you are things that those microbes can do. And in fact, another thing they can do is they can prevent microbes that would cause harm from establishing, colonizing in our body. And so they're really important for maintaining our health and helping us develop. A lot of work has gone into, a lot of beautiful work by a lot of amazing labs has gone into really defining who is there, describing which microbes are present in healthy people and which microbes are present in ill people and which microbes are present in the very early stages of our lives and which microbes are present when we get older and how those microbes are changing over time and with their diet and et cetera. But what I'm really interested in is taking that information of who's there, but then also figuring out what happens when you disrupt those microbes? What happens if you kill some of those microbes off? What happens if you are exposed to contaminants that allows some microbes to grow and kills others? So we do this in our lab. And we're also interested in this relationship between the microbes in the gut. So here I have my beautiful gut that I drew during a seminar, much like this one, given by somebody else. And this is a pretty cool cell layer because I'm just showing it as this little homogeneous cell layer, but there's actually lots of different types of microbes there. And that's something that I studied when I was at the University of Oregon. And there's also this really nice mucus layer, and that's being studied by a lot of great labs, including my former postdoc lab, Karen Gilliman's lab. But what I'm really interested in is who's here in the gut microbiota. This is the lumen of the gut. It's basically a tube, and there's a ton of microbes there. And then how are they interacting with the cells that are in the gut and these immune cells? So this fabulous drawing is of a dendritic cell that's sending a little part of itself through the cell layers to get a taste of which microbes are present in the gut. And then they send a signal inside that says, hey, we need a lot of reinforcement here, and recruits a lot of immune cells, or says, hey, everything's good, and suppresses inflammation. That's no oversimplification, but hey, I've got 25 minutes, not a whole semester. Okay, so this relationship is complicated because there's lots of different cell types, there's lots of different microbes. And then just to complicate things a little more, there's a bunch of genes that are involved in all this too. So you don't have to memorize these genes. I don't memorize these genes. You don't have to memorize these genes. But these are all the genes that are, or some of the genes have been identified to be involved with Crohn's disease and ulcerative colitis and inflammatory bowel diseases in general. And I'm not showing this to freak you out. I'm just showing this to show you that there are a lot of genes that are involved with this complex interaction between microbes and their gut. So what's cool about these diseases, or horrible if you have them, is that they're all characterized by a change in the microbes that are present in the gut and that microbial community and the immune system in the gut. So these diseases are all associated with inflammation in the gut, so an increased presence of immune cells in the gut. And that's also also followed with or associated with painful inflammation, pain in the stomach, change in bowel capabilities, etc. So it's this complicated relationship. And the reason I like to show this image is because there are so many different genes. You know, if you were going to try to study this in a mouse and you wanted to look at the immune-associated genes, let's say associated with Crohn's disease, you'd have to be able to knock out or knock down all of these genes. So what we've done is we've taken a system three-spined stickleback that has a lot of genetic variation, and that just means that there are a lot of genes that are different from one population to another, and I'll talk about that more in a minute. But that's how we get around having to make a knockout out of every single gene in our little host organism. So our lab is really interested in host microbiota studies. We're interested in the inflammatory state and the microbial community, and we do this by making germ-free fish. Now, back in the day, people started looking at, these studies have been going on for a long time. So these are images from Life magazine from the 1950s showing how they would make germ-free animals. So here we have a pregnant rabbit, and she's giving birth in a pool of disinfectant to reduce the number of microbes. And look at this, he's not wearing gloves. This kills me every time. And then here we have another image of somebody who's completely covered head to toe in equipment to make sure that his germs don't get on these animals. And I like showing this for two reasons. One is because I don't have to wear this getup, so we've come a long way since the 1950s. But two, because I like showing this because the animals that they're actually using here are not inbred animals like we typically use now. They have genetic variation. And that's what we're really interested. Because we're interested in this really complex relationship between the inflammatory state and the microbial community and the host genetic background, we like to use animals that are not inbred. So what are we asking in our lab? In our lab, we are interested in how does the host genomic background contribute to host microbe interactions. And we're using this cool little fish here, three-spined stickleback. We also occasionally play with nine-spined stickleback. And yes, to that person in my inbox, I know I need to get you some nine-spined stickleback. But we use this animal, three-spined stickleback. And one of the main reasons that we use this organism is because it has a lot of genetic variation. And in fact, the genetic variation between one population of stickleback and another population of stickleback is the same as genetic variation between one population of humans and another population of humans. So for all of you NIH program officers listening out there, it's a really great model for host microbe interactions that reflect humans, because it has the same, this organism has the same genetic variation as humans. We are located here in Anchorage, and in Anchorage we have access to anadromous fish, and you're going to hear about this a lot. So it's basically a fish that lives out in the ocean, and then it comes into freshwater to lay eggs. And then those eggs turn into juveniles, and eventually those fish go back out into the ocean, spend most of their time there, and then come back in. So anadromous just refers to living part of its life in the ocean and part of its life in freshwater. And then we also have freshwater populations. And these are populations of fish that started out as anadromous. So anadromous are the ancestors of freshwater fish, but then they got caught up in this fresh body of water somehow. Maybe a glacier receded. Maybe they were picked up in a net and dropped off in the lake. In whatever case it may be, they had to adapt to this new environment that was, they were in a high salinity, now they're in a low salinity. They had to adapt to new predator, new prey, new food sources, new pHs, new chemistry, water chemistry, just lots of different things they had to adapt to. And they did that all over Alaska, all over Oregon, Japan, Netherlands, just all over the Northern Hemisphere. And I'm just focusing in on Alaska because that's where we are, and those are the fish that we play with. But if you go to a Stickleback conference, you get to hear about people using fish all over the Northern Hemisphere. Now here we are in Anchorage. We happen to be within driving location of a lot of lakes, and I like showing this one because this shows you all the little blue dots, and those are all lakes that we have access to, and a whole lot of those have stickleback fish in them. So what we tend to do is we drive from Anchorage to grab some anadromous fish from somewhere in here. Sometimes we get them from somewhere in here, and I'll show you that in a little bit, and then we compare those anadromous fish to freshwater fish. So you'll hear those terms a lot. Another reason why I like playing with stickleback, or studying stickleback, is because you can actually watch them develop live. And so what I have here are stickleback in chorions, and this is just watching a live fish through a flask. And what you can see here is the fish. Here's the fish egg. Here's the spine of the fish. This little thing is a beating heart. And here I'm just going through the microscope and showing you there's the tail. You can watch pigment appear. You can watch cells. Depending on the microscope you have, you can actually watch heart beating. You can watch blood flow. And you can watch this over time. So this is 3 days after we fertilize the eggs. That's 3 days post-fertilization. Here's 6 days post-fertilization. And at this point, you can see the eyes are a little more developed. There's more pigment. You can actually, in this video, I was able to show the blood flow. So if you look closely here, I'm going to zoom in. There we go. You can actually watch the blood flow over the egg yolk sac here, and then also within the fish. And again, there's the heartbeat. So you can watch these fish develop live over time. Oh, that one moved. They're alive. they're definitely moving. The other, and I spend way too much time doing this. But the other cool thing about that is if you tag these cells with some fluorescent protein, you can see where those fluorescent proteins are, those fluorescently tagged cells are going. And you can also watch the stages of development. So you can say, you can record when the eye appears, when pigment appears, when the vertebrae appear, etc. And I'm showing you 3 and 6 days post-fertilization, but we can watch this all the way from the single cell stage all the way through to about 3 weeks post-fertilization. We do that a lot in our lab. And this is just showing you that time series. Here's 0 days post-fertilization, 0 hours post-fertilization for age, all the way to 8 to 9 days post-fertilization. And as I said, we can take this out to a couple of weeks. We also get large clutches from single crosses. So if we take one female and we squeeze the eggs out of her, and we can do that and keep her alive, we get 200 eggs sometimes. And then we can cross them with, we can do an in vitro cross in the lab or in the field, and with sperm from a male, and so we can do these really nice genetic crosses. The juvenile are optically transparent, which I just talked about. The evolution and development is thoroughly described over well over 100 years. It shows a lot, it shares a lot of pathways with involved in host microbe interactions and immune responses with mammals, including humans. So this is a really nice series of studies that was done by David Kingsley et al. That you should look up if you're really interested in this. And what they showed is that a gene that was responsible for a phenotype in Stickleback, these number of bony plates here, was also responsible for the regulation of that gene, was also responsible for the regulation of genes in humans that led to us developing the human foot shape that we have from a chimp foot shape, which is really cool. And again, our fish have natural genetic variation. I'm working with wild populations, so I go out and grab fish from the wild and bring them in the lab and play with them. It's really fun. We have lots of tools to examine host microbe interactions. So we have all the RNA, DNA sequencing tools, and in fact, some of these were developed in Stickleback. So we've got some really nice tools to look at RNA and DNA. We can plate microbes, which we do a lot in our lab, so we can play with them later. We can count immune cells. As a postdoc, I counted them this way, which is long and painstaking. So I had a postdoc for a while, Lucas Kirschman, who's now in Kentucky, and he developed flow cytometry. And this was using a protocol from Natalie. Oh my gosh, Natalie. I am blinking on your last name. Natalie, who just started her own lab and is amazing, and you should talk to her, and I just completely blanked on her name. I'll have to look it up later. Anyway, Natalie developed a flow cytometry assay to count Stickleback cells, and we use that in our lab. And then I had postdoc Emily Leskak, who was amazing, and she had previously studied behavior in Stickleback, and so we adapted her behavior assays for juveniles. Okay. So, part 1, let's talk about who's there. In the corner of each slide, you're going to see a list of names, and these are the people who are working on these particular projects. So Ryan Lucas is a master's student in my lab currently. He's wrapping things up. Here's Emily Luskak, who I told you about before. Katie D'Amelio was a lab tech in my lab who is now working, well, she's in negotiations for a job with a government agency, and she was amazing. And then Patrice Timmons started out as an undergrad and is now a postbac volunteer in the lab. So what they were working on is comparing microbes in the water to microbes in our fish. For those of you who aren't microbiologists, most of the microbes we deal with are not this big. I did that just to illustrate. And here I'm showing a fish that has a lot of different microbes, and those microbes most likely came from the water. And Ryan is comparing the microbes in the water to the microbes in the fish. And this is just to show you there's a whole lot of microbes in there. So he's doing this not for one population, but actually, what are we up to? 6 different populations over time. So this is a longitudinal study as well. He has that DNA, and it's sequenced, and I was hoping we would have pretty graphs to show today, but well, you know, bioinformatics, you always run into issues. So we're hoping to have that information soon. But these are just the places that we've gone to. Again, these are all within driving distance. I think Big Lake is about an hour and a half from our campus, which is right here. So these are all places that we go to on a regular basis to grab fish and compare the microbes in the water to the microbes in the gut. And we also go to some other places. So this is a series of lakes up near Talkeetna, and they are really neat because they are, some of these are connected, and there's likely gene flow between the populations, and others are isolated. So we have a series of lakes that are in an area that should have some similar microbial community structures, but the populations of Stickleback some have gene flow and others don't. So it's going to be a really fun series to look at. This is just showing you what this looks like. So we go out in the field, we grab some fish, we dissect them in the field often. Sometimes we take them back to the lab if it's below 0. And then this is showing the traps that we throw into the lakes. This is Ruth Eisenberg, a former student in the lab. And then we take the fish and the microbes and everything else back into the lab, and we do a bunch of bench work as well. So we do a combination of field work and bench work in our lab to look at these host microbe interactions. So Emily and I are really interested in figuring out, because we're doing wild work and lab work, if we could recapitulate the wild microbiota in the lab. So we did this fun little experiment where we looked at the microbiota of adult wild fish. Each one of these bars is a different fish, and each color is a different phyla that was identified in the gut of that fish by 16S sequencing. And you can see here that there is some variation from fish to fish that's totally normal and is what you see in every wild type of animal out there that's ever been sequenced. The other thing to note is that this particular group of fish has a lot of different microbes that are found in humans too, and I'll talk about that in a minute. And then we took our juveniles and we reared them in water that was spiked with water from the wild. And what we found is that at, I think this is 28 days post-fertilization, it might have been 2 months. I'll have to go back to the data and look. But these juveniles had very similar looking gut microbiota to the adults. And in fact, we ran some stats. I'm just showing you richness here, but Emily ran a bunch of stats and showed that there was no variation in the diversity